That's like that.
And I guess we go by
For sure you can do it.
I think yeah, they need here.
Okay.
All right, everybody, I'm going to use the microphone for the Zoom folks. Welcome to those of you on Zoom. Oh, it is tracking me. The camera's tracking me. That's good.
Doesn't work.
Welcome to all of you here to the PhD defense of Connor. I'll make a general introduction, a brief introduction, and then we'll take Connor will do his presentation. We'll open the floor for public questions from everybody. We'll probably then take about a half-hour break. We'll retreat to my lab conference room where the thesis committee will meet with Connor, discuss his thesis, make any final suggestions for improvement if that's possible. And then probably maybe by 1 o'clock or so, there'll be some sort of a reception. I've been told an ice cream cake is arriving with other snacks. We have some champagne, we have some goodies to welcome Connor to the PhD realm here. And before I begin, I want to thank Connor's thesis committee, which consists of Professor Stephanie Pierce, Professor Andrew Wiewiner. Andrew, where are you? Oh, back there. Good. Thank you. And then let's begin. So Connor came to us in 2019, undergraduate degree at William and Mary, Cal State Long Beach, master's degree. I couldn't print out the whole CV, but there were 30 publications, or getting close to 30 publications to date. Since he's been here at Harvard, Connor's been a tutor at Lowell House with Megan. He's shortly after leaving, he produced Offspring, who is attending the seminar. He received multiple teaching awards for his work in LS2, something you may rather wish you hadn't had to do, but LS2, the biology of fishes class. And then in the lab, Connor's been a fantastic lab member. R coding, just fantastic skills there, helped so many other people with so many projects in the lab, and of course, used his skills to conduct his thesis research. Now, I want to say just a word about Connor's thesis research before we hear him talk. I think his thesis is particularly impactful because it really brings together two fields that have largely been separate. So for my whole career, we've been working in the laboratory to understand fish biomechanics, aquatic propulsion, principles of hydrodynamics, kinematics of aquatic propulsion in fish. And we're working in a pretty small setup with smaller fish doing highly controlled behaviors that we manipulate the best we can, but we've always known this isn't really fully representative of what fish are capable of doing in the field. And on the other hand, there's this huge developing field of movement ecology, where people are attaching biologging tags to animals, terrestrial, of course, birds, but also in the aquatic realm. There's a tremendous amount of interest in understanding what animals do in nature as they migrate, as they interact, as they reproduce, what types of movements do they exhibit, where do they go? And yet that field has been more ecologically focused, behaviorally focused on trying to understand sort of big patterns of animal movement with not much of a focus on the specific kinematics, biomechanics, how do they interact physically with their environment. Now, Connor's thesis brings both of these areas together in a beautiful way using biologging tags to really inform us about what animals are doing in the field from a biomechanical sense. So I think it's going to be a very impactful set of papers that come out of Connor's thesis that will have a tremendous influence both in the world of sort of more laboratory-based biomechanics and in the more sort of field, ecological, behavioral area of movement ecology. And by doing that, I think your thesis is going to be tremendously impactful. The first chapter has been published. The second chapter got very good reviews back recently. We have 2 more chapters to be published. which I'll be working with Connor on, of course, over the next month or so. But I think at this point, it's good just to introduce Connor, welcome him to his thesis defense. Thank you personally so much for everything you've done for the lab. For me, I've learned so much from you and from your approach to understanding animal locomotion. It's been a tremendous pleasure.
All right, everyone, thank you all for coming. And thanks everyone attending on Zoom. And so, as George mentioned, I'm going to be talking about this pipeline of moving from the lab into the field. And so when we think about how animals move, we think about it on these huge spatio-temporal scales. So you can think of Arctic terns here that move from the Arctic to the Antarctic and back every year in the cycle. So they're moving huge distances. Or you can think about an animal more close to my heart, white sharks, right, which spend half their life basically in these coastal nearshore environments and then take off and move into the middle of the ocean, thousands of kilometers away, and move back again. And so when we think about movement, we think about it on these large scales and how animals move and where they move. But when you think about movement, you can kind of think about it as one continuous track from when you're born all the way until when you die, right? You can think about how you've moved through space continually through time. However, you can kind of zoom in from these large scale processes into like mesoscale processes, thinking about how you can search for food, your patterns of movement during you're searching for food, to these more acute things like, well, you either capture food or escape a predator. And you can think about these really acute things, like each individual step you make actually accumulates over time, and it's actually generating this lifetime track. And so when we think about this movement path, we are thinking about it in this spatial context that's rich with environmental stimuli and interactions with conspecifics. However, it's really dictated ultimately by how you move, right? How each step you take, how fast you move, which direction you move, right? And while we measure movement path out in this environmental context, we really often measure how we move in a very different environment, right? We bring animals into the lab in these really highly constrained contexts and basically put fish in boxes and then introduce some stimuli like flow and then measure how they swim. We can basically take videos and you can really, really accurately measure how different parts of an animal move through time, and you can reconstruct its locomotion. And I think this is really powerful as a tool, and it's taught us an incredible amount of knowledge about how animals actually move. However, this is a very different environment than the rich, diverse environment that you'd see in the wild, right? Where you have vertical movements for animals. However, in the last 20 years, we've kind of begun to bridge this gap a little bit. So animal-borne biologging has really taken off. And so the idea is, instead of bringing animals into the lab and recording video of them, we can attach little data loggers onto an animal and then release it into the wild, and the tag records what the animal is doing through time. And so this is like a classic paper, one of the early ones, where they put tags on penguins, release the penguins, and you can differentiate walking versus tobogganing. So that's when they're on their bellies and kind of crawling along. And what these tags do is basically, at their heart is an accelerometer. And so I think the easiest way to think about an accelerometer is basically a ball suspended by two springs in a tube. And if you shake that tube, you can think that ball is going to compress one spring or the other. And that's what the tags are actually measuring, how those springs are compressed. And so these tags tell us tons of information on things like frequency of movement, the orientation of the animal, like the posture. So you can see like laying down versus standing up, as well as the intensity of movement. So how intense are the accelerations of the animal. And so this has been taking off, and it's really focused on this kind of the behavioral context, this ecological context of how animals move and what they do in the wild. And so this is really where my background came from. And so when you do this, you often take one tag, place it on an animal, and then infer the entire animal's movement and behavior from that one location. However, I really got into kind of this biomechanics thing, because when you tag an animal like this, I really wanted to kind of remake these animations to animate how the whole animal moves based on that one location. And you really can't do it, because you don't know what the head yaw, the head amplitude is, or the tail amplitude. And we know that this is different depending on which species or style of locomotory gait you can use, right? Where different species have different tail amplitudes, different head amplitudes, and different wavelengths. And so kind of this is like a fundamental limitation of a problem. But however, you can also alternatively think about it as just like it's a data limitation. Instead of just using one tag, we can actually place multiple tags on the same animal. And then you can use those to understand how the different parts of the body are moving at different times. And you can use this to basically reconstruct how one of these midlines looks through time. And this is basically the crux and core of this entire PhD, is trying to move from this single tag into integrating these multiple tags to recreate midlines. And so just to give you a quick outline of what my PhD is. In chapter 1, we're going to talk about how we can ensure different tags across the body are temporally synchronized. In chapter 2, we're going to talk about what can be measured and how accurate are we when we actually measure it. And then we're going to move into some more biological questions, thinking about how does the head and tail of the shark move? Do they move the same way or do they move differently? And then finally, we're going to kind of even go out even further and kind of just generally think, how do sharks swim in the wild? We're going to go try and measure that. So chapter 1 is how much the tags drift and can we correct this? And so we can think of a human walking like this, right? And so here's this nice little stick figure walking along, and you can have a black leg and a red leg, right? We can put a tag on the individual's black leg, and you can measure some metric like this. You have an oscillation in leg speed. Imagine you're on a treadmill. This isn't very accurate for actual walking locomotion, but it gives you an idea. And we're going to see a lot of graphs like this in this presentation, so I just want you to get familiar with it. And so anytime this line is above 0, you can think of the leg is moving forward. Anytime the leg is moving backwards on the treadmill, you can think it would be negative. We can put a tag on the individual's other leg, the red leg. You can generate a graph like this. And so you can see the black legs and red legs are moving in anti-phase, right? When one's moving forward, one's moving backwards. And so we can basically, by looking at these patterns between the two tags, you can infer how the individuals are actually walking. We can contrast this with a behavior like this, which you might think of as hopping, right? Where your black leg looks the same, but now your red leg is actually in phase with your black leg. And you would interpret this as like you're hopping along in movement. And so we can think about the time difference between these two tags is really important to understand the relative timing of movement. And the problem here is if these different tags are moving at different times, or at the time the tags are recording at different rates from each other, it'll lead to drifting. So maybe you start walking normally, and then as you're walking, these tags drift apart, and then your legs become out of sync. And so if you actually look at the human walking, right, these legs are slowly drifting, and then your biological interpretation of how that individual is moving is very, very different than how they actually walk in the wild, right? And you can see how these signals drift. It looks like his legs are spinning around, but they're walking. And so the question really becomes, do these tags drift from each other? And what is the magnitude of it? And so to do this, we can basically take a bunch of these tags, strap them to this rod, and then oscillate this rod back and forth. So each tag is experiencing movement at the exact same time, and we know that. If you do this at time 0, all these tags are nicely synchronized with each other. And if you do this again 24 hours later, you'll see that now these lines, right, these colors have all shifted from each other. You can measure this amplitude as 0.2 seconds or 0.45 seconds. And these can be kind of significant when you try and interpret the biological locomotion of the animal. And so the question becomes, we have these 2 points, but what is the pattern and drift over time? Is it nice and linear like this? Or do we have some sort of complex pattern that would actually be much, much harder to correct? And so what we did is we flapped these tags every 30 minutes through time. And basically you see these drifts are really nice and linear, which is great because it's really easy to correct them. However, when you look at different batches of tags, you can see very different magnitudes of drift. So this is actually a different batch of tags. And the scale here is different. So these tags drifted by like 6 seconds from one tag in just like 9 hours. And so now not only are you like drifting within a step, now the tags are saying different steps are actually happening at the exact same time. But again, it's nice and linear, so it's easy to correct. And the best part about it actually is if you do this experiment repeatedly through time, you'll actually see the drift is repeatable. So basically, when you test the tag again, it has the exact same drift rate. So basically, each tag has a characteristic drift rate that you can use and then predict into the future. And so you can measure these things in the lab and then use these tags in the field and correct the drift post. And so that's kind of the crux of this chapter. And it's kind of dry, but what I want you to take away is that time synchronization here is really vital. It's at the core of how you interpret all these signals. And in these experiments, we show it's linear and repeatable, and this means you can easily correct it. So if you have a red signal like this that's drifted from its true, where it should be, the blue, you can basically go through time and pull that back to where it's supposed to be, which allow you to easily or accurately interpret the biological context. And this is great because this allows us to use these kind of like relatively cheap and independent tags and kind of place them wherever you want on an animal. So you can kind of think of it like motion capture, right? You can place a tag somewhere and you can measure how that body part moves without having to design an entire custom tag that measures that location and is connected to some other brain. So if we can do this now, the question then becomes, what can we calculate using this method? If we use multiple animals or multiple tags in the same animal, how do we combine this to create these kinematic parameters that we care about? And ultimately, what is the accuracy? Not just can you do it, but is it real? And before we get into that, I want to talk a little bit about aquatic locomotion. And so you can think of some eel swimming like this. If we just take a snapshot of it, we're going to kind of calculate some important parameters. So you can think about amplitude of the body is like how far you are from some general midline. And you can think of wavelength along the body. So this is how long this wave is as this animal moves. And you can use this to characterize the locomotion. You could compare this wavelength to a wavelength of 0.5, right? So this is a shorter wavelength. There's more waves within the body. Or you could think about a wavelength of 1.25, where actually that locomotory wave is actually longer than the animal. And so you can kind of think of this wavelength parameter really dictates what the locomotory style looks like. However, in all these contexts, they have the same frequency and amplitude. However, we know that the head amplitude for most fish is much smaller than the tail amplitude. And this is using the same wavelength. And so instead of thinking about it, we can actually, instead of thinking about it in this spatial context where you track the whole animal, we can just place two little dots on this one fish. And instead of thinking in this spatial context, we can now turn it into the time domain. So if we track these points, the lateral position of these points through time, you can see the green line has this lower amplitude and the purple line has a bigger amplitude, which makes sense. If we track, the peaks of each one of these waves, you can estimate the period. And then if you track the difference from when each peak occurs, you can calculate the time lag between these two signals. And the reason this is important is because if you go back to like the formula for wave speed, you can use this to basically back out what the wavelength is just from this period and lag component if you know the distance between the 2 points on the animal. And so basically just using two tags, we can estimate what a wavelength is. And so this is the theory behind this. How does this actually work in real life? And so to test this, we use this, we put a bunch of tags in this nice silicone body, and then we flapped it. And so we move this silicone body back and forth at different degrees of frequency, yaw, sway, to try and kind of create a diverse sample space. And then we can basically take a high-speed video and shoot it from the ventral side so you can see how much the body is bending, and we can track each one of those tags in this video. And so this allows you to estimate the lateral location of each tag, as well as the orientation or the heading of each tag from the camera. And so we can now compare what this IMU data looks like to a camera tracking data. So the bottom here is just data from the first tag, this blue line. You see this nice oscillation over 2 seconds. You can compare it to the top, to the heading data from the camera. They look the same, which is good. However, we don't just have this one tag, right? We have multiple tags, so they're all lined up temporally. And you generally see this high agreement. When you look at these two graphs, they look the same. We can calculate the amplitude of these two waves. If you plot them, the x-axis here is your camera, your y-axis is lateral amplitude of the tag, from the tag, and you'll basically see it follows along this nice one-to-one line. And we can do the same thing with phase lag. And so this is really nice, but now this is just from one trial at one set of flapping conditions. We can plot this across a whole series of different flapping conditions. You'll generally see there's pretty high agreement. There's a little bit of bias here, but there's really high agreement between what the camera is telling you and what the tag is telling you. And so this gives us really good confidence in what that we're able to recreate these metrics similar to how a camera does. And if you actually export this back out to wavelength we talked about, we can actually measure what the wavelength of that flexible body is. And we can see that basically as your flapping frequency increases, you flap that flexible body faster, the wavelength of the body decreases. So at slow frequencies, you can think of this long kind of sinusoidal wavelength. And so this is really nice. But this is like a pretty controlled setting, right? Where I like have these tags each other in this controlled laboratory setting. They don't, nothing changes. So the question is, how does this work on real fish? And does this work when you really push the limits, right? When you go to this really high performance criteria that we are interested in for biomechanics. And so to do this, I worked with Ted and Elsa, and I went to the Conti Lab, which is part of the USGS, and they have this incredible facility where they have a humongous flume, 20 meters long, in this big holding tank. And so what we did is we tagged white sucker with 2 tags here, and you drop them into the holding tank, and the fish will then burst and swim up the flume whenever they want to. You just let them do it whenever they want to on their own time. And what this looks like is something like this. You can hear those little taps. That's actually the tag hitting the glass when they're swimming really, really fast, to give you an idea of how fast these tailbeats are, right? And then the fish can't get out the back, so it actually comes back down the flume backwards, right? And this, of course, is too fast to record using a regular camera. So we can actually put high-speed cameras using a ventral mirror and film how the fish swims up that flume. So you can see 2 tags right there on the fish, and here it is swimming up the flume. And this water flow is like quite fast, like 2.5 meters a second here. And so we can do, again, we can take this, and we can track multiple points on the fish's body, and 2 of these points roughly correspond to the location of the tags. And so we can, again, plot this, like I've been showing you, where you have lateral position of each tag, and this is based on the camera data, over time. So it's over 2 seconds. Each one of these oscillations is a single tailbeat. We can compare what this data looks like, or you can see that the posterior has a much higher amplitude than the anterior of the body, the mid-body here, orange. You can compare this to what the tag data is, and generally you see the purple spikes are much, much higher than the orange spikes, right? And so you're recapitulating, you're recapturing that information of the relative motion of the body. Keep in mind that these units are different here. So the top here is gravity, and these are really high accelerations, right? The lateral accelerations you're experiencing on the caudal tag are like 30s, like when you talk about pulling G's on like an airplane, like this is 30 G's. I mean, it's not sustained, so it's a little bit different, but they're quite intense accelerations. So this is great. How do we compare this? All right, so we can basically go out and look at how does each method compare frequency. And so we can see that generally the black line here is the camera, the green line is the estimate from the tag, and we'll see that these things basically overlap. They give you the exact same peak frequency, more or less, or average frequency, which is like 12 hertz, which is like really fast. So 12 hertz is like the maximum wing beat frequency of a pigeon. And you can think of a pigeon flying pretty fast. And they're in air, not water. And so I think this is pretty impressive. However, we can then use these same time difference estimates and measure wavelength of the animal. And it works pretty well. And so generally the black line here is from the camera, the green line is from the tag. And you can see this error bar, this shaded area here is actually the kind of the confidence interval around our estimate. And that's because these tailbeats are happening so fast, we actually only characterize them using 8 points. So you actually have relatively low sensitivity to look at temporal shifts between them. But generally speaking, these estimates of wavelength are the same. And this is estimating wavelength at really at the maximum, like the top end of what we can do. But the coolest part of this tag data, right, is if we zoom out here, this is what is captured by the high-speed camera as the fish swims by. The tag data is unbiasedly recording the entire time period, and so we can zoom out and we can really characterize the entire burst. We can see as the fish is swimming in the holding tank, it has these inconsistent tail beats. As it swims up into the flume, it beats its tail around like 4 hertz, and then you can characterize the total burst. So how many tail beats there are, what's the maximum frequency. And I think this is really kind of extending this idea of how we can measure what this like maximum performance of fish is. And then you can also then, of course, see after the burst has ended and it starts swimming back down the flume with a tail beat frequency around 3.5 hertz. And so I think this really highlights the power of using this type of methodology where you can recapture many of the same kinematics you would get with high-speed video, but you're able to do it over much longer periods for actually relatively cheap compared to a high-speed video camera. And so I think this is a really nice way of looking at this data, and I can show you we can capture these metrics over time. However, I think what's more interesting sometimes is actually recreating how an animal looks through time, right? This is what we want to do. We want to almost recreate what the video data looks like we can visually see how animals are swimming. And so what we did for this, we grabbed a shark, put it in the basement of the MCZ labs, and put a couple tags on it. As you can see, these little white blocks are tags, and then we would record video and let the shark swim for around 24 hours. And I'm not going to show you too much data here, but I'm going to show you some reconstructed midlines. So the graph on your left here is data from the tags that we recreated how the shark looks, and the graph or the movie on the right is data when you recreate how the shark looks from video data. And I think I find these very convincing. In fact, when I first made these plots, I actually thought I was plotting the same thing twice because I think they look so similar. There are a few differences, but I think this really shows you this tag methodology allows you to really recapture a lot of what's happening. Also visually, we like to watch. And so I think just in conclusion here, we can really recapture all these important parameters that we would normally measure biomechanically, like lateral amplitude, heading amplitude, phase lag, wavelength, frequency. And the power is we can do this continuously and we don't need animals to be in front of cameras. And this allows us to move to larger animals and new environments. And so moving on here, we can go into chapter 3. So this is kind of moving out of the basement, I think about it, right? And so instead of using these constrained laboratory settings, we can start thinking, how do animals and sharks really, in chapter 3 and 4, behave in the wild? And so in this, I read this paper by Miriam Porter in 2020 that got me really thinking a lot. And what they did was they flew drones in the wild and then shot video down on shark swimming. And then they tracked the different parts of the body. What they found was the head frequency was faster than the tail frequency of the shark, which I was like, that's weird, and because I'll get into it a little bit, but it's really weird. And then when you dig in a little further, it's been documented in a couple of other species, like bonnethead sharks, scalloped hammerheads, Caribbean reef sharks, and it's largely been and then in a couple of non-sharks as well, sturgeon lamprey, with some ideas that maybe it has to do with like this cartilaginous backbone, they're not bony. But it's really based on this idea that we're seeing this more in like natural environments when you're not in these highly constrained flumes, right? Whereas animals can behave as they wish. And so the reason this is so weird is it like fundamentally breaks what I told you earlier. And so when we model how a fish moves, right, we kind of think of it as this rearwardly propagating wave. And so the assumption here is that the period at one part of the body equals the period at another part of the body. And if you measure this phase lag, you can measure it here. And basically all I want you to know is that basically, if you measure it, you can put this distribution here. The only one of these pie slices is purple here, right? That basically you have one phase lag across the body, right? This is a really narrow distribution. And I actually tried to make an animation with the head and tail moving at different frequencies, but it's actually pretty hard to do. But you can think about it in a walking context, which is pretty hard to imagine. But if your left and right leg move at different frequencies, it looks wild, right? Where you're basically, it's like a hop, skip, and a jump, or a hop, then like a left step, then a hop, and then a left step, then a hop, then a right step. It looks pretty wild. And so I think, yeah, let's leave it there. And so we're gonna bring the same graph here, right? And so you can plot this same frequency of these two body parts, and you have all these purple little boxes here, right? And this creates one little pie slice that's purple. You compare this to the anterior and posterior having a different frequency. You'll see these purple bars have variable lengths, and that basically what this ends up with is that there's a lot more purple around this pie chart, which shows the phase is constantly changing. And so what you just need to know is that these two things look different. If you have the same frequency, you have one pie slice. If you have different frequencies, you'll have many pie slices. And so how do we actually get the head and tail frequencies of animals in the wild when they're volitionally swimming? To do this, I created a new multi-tag package. I just basically took these little data loggers and I would suture them onto sharks. And then the tags were actually held in place by these things called galvanic timed releases. And so you can kind of see one right there, and that's a tag right in the caudal fin of a shark. And the way these work is basically there's two eyelets held together by a piece of metal. And when you put that piece of metal in seawater, it'll slowly corrode over 12 hours. And once it corrodes, the two eyelets are detached and allows that tag to detach from the animal and float to the surface. Or there's a little VHF pinger, and I can go get it. And so when you tag one of these sharks, you can release it into the wild, like here. This was in Yarmouth Port up by my house in the Cape. And so you can see each tag on the body, and there's this big orange thing, which is the float. This is actually the smallest shark I tagged, so the tags look a little big, but not too bad. You can easily see as each part of the body moves, there's different amounts of movement. The tag pops off, and you have to go find the needle in the haystack. So there's a little tag right there. And luckily, I was able to get back all the tags I put out, which is great. And when you plot this data, there's a lot going on here, but when you plot this data, at the very top here, you see there's a green line oscillating back and forth. That's the anterior. There's an orange line, and we can see that basically the frequency increases as well. But you see that the frequency is, there's very minor differences between the anterior and posterior frequencies. And I don't have a pie graph here for you, but if you look at this purple line, that's the phase difference between the two parts of the body. And you'll see that there's very little variation in the phase difference through time. And so that means these things are like phase locked together. We don't see any of this dual oscillation thing. And so if you plot this for the entire deployment of the shark over 10 hours for the shark, you'll see that basically only a very few pie slices are filled. And you can do this for all the sharks, and you'll see the same thing is true. And so we really don't see any real evidence of a difference between the head and tail frequency in these sharks. And when you start looking at the video of sharks swimming in the wild taken by these drones, you can start to kind of think about why you might see these differences. And so there's a bunch of water motion as surge moves back and forth. There's a bunch of wave action, right, which is causing, you can see all these little seaweed particles to move, the sargassum. There's all these surface reflections that are causing small scale deviations of how you see the shark. And then the shark is actually turning as well through time. And so all of these things kind of are little small aggregates of error when you start measuring the movement. And if you actually look at one of these tracks from this Porter paper, you can kind of begin to see the data is actually relatively noisy between the different body parts. And it's also really short. So we're trying to estimate these differences in frequency from relatively few tail beats. And I think this is really kind of ultimately the problem, is that when you only are looking in this frequency domain, like you do an FFT, or you compare peaks and directly compare frequencies, it's very easy to have differences in frequency. Like in the study here, it was like 62% of the time or 74% of the time, the head and tail of the shark actually had different dominant frequencies. However, those frequencies were different by a very relatively small amount. And when you actually compare the phase shift, there was no real variation in phase shift, or it was very minor variation. And so I think this kind of gives us a glimpse of how we can take these, how we can use these tags to measure these questions in the wild. And so it's kind of it's a little snapshot. We can move beyond this and try to think of how do sharks actually swim in the wild, right? What do these movements look like as they move through the water? And so we can think of back to these ideas. So you can think of like how much the head of a shark moves, the head amplitude, or we can think about how the variation in tail amplitude, right? Or maybe sharks will have different wavelengths. Which one of these, right? There's a nice little groovy shark here. The pectoral fins are distorted as well as the body. Sorry about that. Or we can think of frequency, right? At what frequency do sharks actually swim naturally, right? And so instead of forcing conditions on the sharks in laboratory conditions, we can let the sharks tell us what they want to do. And so the question is, in the wild, which one of these dancing sharks is really the right one, is the accurate one? And so to do this, I'm using the same data set I just showed you in the last chapter, where I have 5 sharks here because this tag on shark number 2 actually didn't record for me, and I also didn't get a picture of it the same way, but we can still use it. And so the question is, what did these sharks do after we released them in the wild? And so here is just data from one shark. And so this top graph here is depth of the shark. So you see I release it here in about 5 meters, 15 feet of water. It swims to the bottom, and then it slowly swims into deeper habitats until about like 7:30 p.m. And then it takes off from the bottom and swims up in the water column, which I think is kind of neat. And you have these occasional deep spikes to the bottom. At the same time, we can use the tag data to measure the frequency of the animal. And so each one of these points is an individual tail beat, right? And so this is 25,000 tail beats we measured from the shark over like 9 hours or something. And this is just one shark. We can do the same thing for all the sharks, right? And you have something like 100,000 tail beats. This is orders of magnitude more tail beats than you would normally measure in a biokinematic study, right? And so you can see there's some differences. You know, sharks 1, 5, and 2 use these like deeper habitats up to like 15 meters, while you'll see like sharks 3 and 4 never stay relatively shallow. I can't actually say if they're shallow or if they just didn't swim to the bottom. They were pelagic, but it doesn't matter. And we can measure basically tail beat frequency for all these sharks. And so how do we aggregate all this data back together into some metric that we care about that tells us things about the kinematics of these animals? And so you can think of some shark like this, and we're going to measure heading for the individual. And so through time, as the shark swims back and forth, we can measure heading as these nice oscillations. Discretize and measure each one of these oscillations as an individual tail beat. And from each one of these, we could measure things like period, as well as amplitude for each tag through time. However, there's a kind of like, you get a lot of noise in these signals, right? Individual tail beats can have error in them when you measure them this way. What we can actually do is basically combine all these measurements together and then use an average to figure out what does the average tail beat look like at any one frequency. And so if you do this for the tail, we're looking at yaw, so this is degree, like heading of the tail tag. You see there's like about a 40-degree oscillation back and forth. And it's actually quite a nice linear line. And so you just put points 40 degrees to one side and then 40 degrees to the other side. We can look at how that oscillation changes, or the heading changes across the body. You see the mid-body is about 10 degrees, the anterior is about 6 degrees oscillation back and forth, which about makes sense. However, a kinematic thing, we're often very interested in thinking about the lateral degree of ale, has about a 9 centimeter oscillation. And these are about 1 meter long individuals. The mid-body oscillates around 2.1 centimeters back and forth, and then the anterior tag is about 1.2 centimeters. And so I'm going to take the same data here. I'm just going to show it to you in a slightly different context. And so we can take that information and put them all on the same graph, right? Stack them all together, and we can see how each body part moves. You can think of phase differences between these, and here's lateral displacement. And this bottom graph is actually just showing us these top 2 graphs again in a slightly different context, where we have some foils that show you the orientation of each body part through time. And so you can plot this as a nice little movie, and you can see how each has the head, mid-body, and tail move as they oscillate back and forth, and what degree of heading there is as it moves. Of course, I was talking about, there's this all-important lag parameter that's really important to describe how the whole motion of the body moves. And if we can take this, and instead of plotting this as time proportional to tail beat, we can start plotting how the value of each body part. And so you can plot lag across the body, which gives you the slope, which tells us what the wavelength of the animal is. We can plot the degree of lateral amplitude. We can fit through a polynomial. And basically with this, using these two graphs, we can basically infer what the motion is in between the 2 points that we measure. And this allows us to create these midlines of what the whole body looks like. Or you can plot it through time where, sorry, it's so light, but this light gray line shows you what the whole midline of the shark is. And so this is all data just from a single frequency at 1 hertz. These sharks displayed a huge diversity of frequencies as they volitionally swam. And there's a lot going on here, but all I want you to take away from this graph right now is that largely speaking, there's very little difference in lateral amplitude with changes in frequency. As you go from 1 hertz to 6 hertz, we see relatively minor variations in displacement. And the maximum frequencies we observe here are quite fast. So 6 hertz is pretty fast for an animal that's 1 meter long in water, right? To give you a context, Usain Bolt, when he runs the 100-meter dash, his step frequency is like 4 hertz. And so this is quite above that. However, this is all just characterizing the steady state swimming performance. We can also think about sharks don't just swim in a straight line. We can use the same tag data to understand all these really dynamic motions. So we can see here, we have a shark that's swimming in a straight line here, like right, the heading doesn't change. We see this large shift, and then it swims in a straight line again. This is a turn that's about 203 degrees. And so these graphs are a time series of what the midline is. And so at a peak flexion, bending of this turn, its head is almost touching its tail, right? It's really curved on itself. And so these are, I think, pretty important, or pretty impressive. And so some of these maximum turns, like 286 degrees. This is almost a 360-degree turn within one single tail beat, right? We're not doing 2 tail beats to do it. We're doing only in 1 tail beat. And you can go beyond these short things and start scaling up to these behavioral contexts. And so here's a graph of heading of the shark over 30 minutes. And so you'll see there's all these dashes, right, as the heading is changing. You can kind of think of this as circling, since we can take these heading graphs and extrapolate them or dead reckon them and estimate what a track of that animal is. And so you see it swims straight here, and then it does these corkscrews. And so it does corkscrews for 10 minutes. It circles for about 1 meter or 1 minute in duration for 10 minutes, and then it breaks out of it and swims in a straight line again. And so, right, you can think about this turning is actually a pretty behaviorally significant behavior, right? And it's dictated by how the animal is actually moving through the water. And we think about how important is this behavior? How much time do these animals actually spend in this turning mode, which is worse characterized in lab studies. And so we can make a graph like this. And so your y-axis is percent time steady, and your x-axis is some threshold of turning angle. And so if we say, I'm going to say anything less than 10 degrees is going to be steady swimming, and that's what we measure in the lab. You can see that about 65–70% of your time would be dictated as steady. And this is based on an individual tail beat. When you think of like steady behavior, you're often thinking about it in this temporal context, where it's not just one tail beat has to be straight, but you need multiple consecutive tail beats in a straight direction to kind of be steady or stable. And so when you start plotting consecutive straight tail beats, you'll see that 10 straight tail beats in a row, you're down 20% of your time. And so I think this is, a really kind of important thing to think about, is that when we measure things in the lab, we're often measuring the straight line, steady state dynamics. But if you think about it in the wild, animals are kind of rarely doing these straight line normal behaviors. So we have this mismatch between the lab and in the field. And so just really in conclusion, I mean, when you think about how these sharks swim, right? I'm gonna say sharks swim normally, right? Like if you had me guess how a smooth dogfish would swim, I would guess something pretty close to what we ended up measuring, right? Had a wavelength of 0.77, a head amplitude of 0.03, a tail amplitude of 0.22, which is basically exactly what like Webb and Keyes, like one of the foundational shark swimming kinematic papers showed, that like smooth dogfish had a wavelength, or leopard sharks, which are relatively closely related, have a wavelength of 0.77 and a tail amplitude of 0.21. And if we move beyond sharks, we can kind of compare this wavelength to a whole diversity of species. And you can see our value basically falls within the middle of everyone. Or you can compare this with Elsa and Val's paper. Our tail amplitude is higher than what they found, but it's really 0.2 is really right in line with most kind of fish tail amplitude studies. And the head amplitude is right in the middle here. And so I think we're moving from these kind of static conditions, right? And we're able to really accurately measure how these animals move in the wild. And so this is where we came from, right? Putting fish in boxes and forcing them to swim how we want them to, which tells us an incredible amount of information. And you can do things like get metabolics and do muscle physiology, but you're really constrained in what the animal is actually doing. And so in my PhD, I've tried to make this a pipeline to move out of this narrow paradigm, right? So in chapter 1, I show you can synchronize tags. In chapter 2, I show you that these tags can accurately recapture the kinematics of fish. In chapter 3, I show you that fish oscillate with one frequency, which is good because that basically kind of supports how we think fish swim. And then in chapter 4, sharks swim like a fish, but spend lots of time turning. And so I think this is where we're going, right? We're moving out of these controlled laboratory settings and allowing us to tag these huge animals, or like a white shark, or these very large and really high performance animals like bluefin tuna, and allow us to move out of these contexts of steady state swimming to think about things like foraging, migration, as well as moving into kinematics of like new habitats, right? How do deep sea sharks swim? How do sharks in really cold environments like Greenland sharks swim? Moving into habitats like nighttime, measuring kinematics when you can't actually see the animal as well. And this allows us to really ultimately link form and function between these between animals. And so with that, I would just like to say thanks for listening. But before I release you, like I released the shark, I have a bunch of thank yous. So thank you to Stephanie, Andy, and Andrew, my committee members, for always answering any questions and for meeting with me, a bit probably not as frequently as they would have liked. Thank you to Elsa and Ted for, I have a bunch of data that I didn't show here, but for allowing me to use the Conti facility and facilitating me to tag suckers and lamprey out there and get this really cool high performance swimming. Thank you to all the members of the Louder Lab who have helped me with either field research, tagging sharks, or just talking and thinking about how we can measure fish kinematics through time. Thank you to Lowell House and Winthrop House as well for 1 year. Help for kind of providing me a home and really a community through my entire PhD, where you felt like I always could go and interact with people and have a really welcoming, nice environment. Yeah, I know, Freddie. So it's done. Thank you to my family for always being super supportive of everything, for being supportive of me and encourage me to always do whatever I want to do. Finally, I'd like to thank Nick Whitney here, who actually got me into tagging sharks originally 12 years ago, and also introduced me to Megan, my partner, when I met her shark fishing when I worked for Nick, who has read every document I've ever written, has been providing unending support and encouragement to me, and even probably got me, and ended up with me in George's lab. And finally, for producing Freddie, I'd like to thank you. Freddie, while you probably hindered my PhD finishing, you definitely made sure I was up many nights, including last night, thinking about my PhD. And finally, of course, this wouldn't be complete without thanking George. George has, thank you, George, for taking me on as a grad student with very little kind of biomechanical background, and encouraging me and giving me the space to do these things. So George loves getting his hands dirty. And so here he is helping me tag this shark in the basement. I didn't get him on a boat to tag sharks, but I did get him out into the field tagging suckers. You can see George here on the step ladder, acting like a bird, trying to scare the suckers so they'd swim up the flume. And I think, and so George is just like an incredible resource. And I think George is kind of like the Energizer Bunny. He is just like infectious with the amount of energy he has and brings to everything. And he really, and the carrot is true, where George, there's no stick. He's just enthusiastic and just pulls you along with his own enthusiasm. And it's really incredible. And I think the last thing that I want to say about George is, I don't know when he sleeps, because regardless of when you email him, he responds immediately. I mean, it is a truly admirable characteristic that I aspire to in my life. But he will always respond to you, give you incredible feedback as faster than you want it often sometimes. And so with that, I'd just like to say thank you again all for listening.